Hello, and welcome to INCSD, Innovative Clinical Study Designs. My name is Luis Angel. I'm a research scientist statistician and vice president of INCSD. Today's presentation will be led by our CEO, Dr. Luis Rojas, and together we will be taking a deep dive into the fundamentals of the scientific method and ICH E9R1. As you go through the video, feel free to pause and check out some of the links in the description to learn more about the contents of today's discussion.
I will be focusing on refreshing some key terms, axioms already established for many years in the scientific community, and will share some basic concepts, which I hope will provide clarity in how the principles of the scientific method should align with the ICH E9R1 guidance and its implementation, and the ICH M11 draft guidance, both aiming to promote consistency and efficiency in the development, amendment, review, conduct, and closeout of a clinical trial and the exchange of protocol information. So, with this in mind, let's get started in finding a common ground between the scientific method and ICH E9R1. First, we'll go over the scientific method, its general approach, explain what is and what is not considered in its definition, and discuss the scientific method in the clinical research context. In the second part, we will talk about the operationalization of the research question, a very important term in scientific research in general, and how it is described in the FDA drug development process. Thirdly, we will discuss hypotheses, a key component in the operationalization of the research question. Finally, we will discuss other important definitions like the study aim, objective, and endpoints, the scope and limitations in clinical research, and briefly describe the predominant philosophical epistemological style in clinical research before we start connecting the scientific method with ICH E9R1. The scientific method, in general, is a set of principles and procedures for the systematic pursuit of knowledge, and it consists of the following steps: formulate a research question, run a background and literature check. For example, in clinical research, it will require to fully understand the conceptual domain of the therapeutic and disease area, the competitive landscape, meaning past and present clinical trials, treatment option available, just to mention a few. Then define a hypothesis, which then of course require defining objective, the endpoints to later evaluate and or to test the hypothesis. Keep in mind, not all clinical trials imply hypothesis testings, since some could be descriptive in nature, for example, an open label or a natural history study. Once the data is collected from the experiment, it is analyzed and the results are summarized. The final steps consist on reporting the results and, of course, evaluating the next step regardless of whether the hypothesis was accepted or not. If you were able to demonstrate that there was enough evidence to accept your hypothesis, then the study team could decide to pursue further research into different indication or different patient population, or to explore the long-term safety or efficacy profile of the investigational medical product. Likewise, if the study failed to achieve the evidence required to reject the null hypothesis, additional work may be conducted to, for example, explore the efficacy and safety of the IMP in a different patient population, subgroup, or maybe in a different therapeutic area. If that is the case, then a different research question could be formulated and the process will restart. Let's evaluate for a moment the epistemological frame associated with clinical research, meaning the reasoning style that is almost mandated by default, predominant, which include how we will approach the theories and methods, the route that we will take to perform our analysis, testing, and to evaluate the results. The answer is quite straightforward, and the epistemic style in clinical research is called positivistic empiricism, which states that true knowledge should be based on observed facts gained through scientific investigation, following very well-defined processes and procedures and with validated instruments, in where bias and data integrity are properly controlled. This philosophical system holds every rational, justifiable assertion which can be scientifically verified or is capable of logical or mathematical statistical proof, and therefore rejects metaphysics and theism. The system confines itself to the data collected and excludes any a priori or metaphysical speculations. So now we can affirm that the scientific method is about following a systematic process for observation and experimentation using either inductive or deductive reasoning or a combination of both. But what is not the scientific method? For starters, the actual aims and products of science, such as knowledge, prediction, or control. Methodology, which include the values and justification behind a particular characterization of the scientific method, for example, the methodology itself. Values such as objectivity, reproducibility, simplicity, or past success. The method itself is distinct, to some degree, from the detailed contextual practices through which methods are implemented. Contextual practices might range over, for example, science-specific laboratory techniques, statistical formalism, or other specialized language used in description and reasoning. Technological or other material means, ways of communicating and sharing results, whether with other scientists or with the public at large, or the conventions, habits, enforced custom, and institutional controls over how and what science is carried out. In the overall context of the scientific method, we have the method itself, which could be motivated or justified somehow following some methodological rules, which is the system designed to develop and test a methodology for a specific, definable purpose, and as such, to provide for the development of and the research into methodologies. These methodological rules are proposed to govern method, and it is a methodological question whether the method obeying those rules satisfies the given rules. Now, let's see the scientific method under the clinical research context, and keep in mind the 6 common steps previously outlined on slide 3. We begin with the formulation and operationalization of the research question. More on that later. And once we have our research question, we proceed with literature review to define not only the conceptual domain, but also the regulatory requirements, and the background check, which include the disease characteristics, previous studies, and other elements that could explain the mechanism of action of the experimental treatment if it is known. Then we move into hypothesis generation process and define the trial objective, endpoints, and limitation, which I will also explain later. At this stage, we also draft the research procedures and assessment, the schedule of activities, and almost in parallel, we determine the feasibility of the trial from the subject, study participant, operational, and financial perspective, and many other technical requirements for the execution of the study. The draft protocol is written, and after further calibration, the final protocol is released. The sites are activated, and the subjects are screened and enrolled into the study. It is also expected that during the execution of the trial, the implementation of strategies to minimize bias, such as randomization, blinding or masking, and or control of confounding, could also take place. With the trial now up and running, trial data and method are reviewed during interim analysis. This will take place with or without the intervention of the data monitoring committee. Modification and adaptation may also occur, depending on the nature of the study design, or if the study requires modifications. Then possible amendments will take place before the database is locked. After database lock, the final analysis is completed and the results are published. a very simple explanation for a very complex process. In this presentation, we will cover only the topics in the blue box, but stay tuned as we develop plenty of material to cover all other components with a special focus in biometrics-related activities, meaning statistics, statistical programming, and data management. Let's move on and talk about the operationalization of the research question and the W questions that need to be answered in order to define elements required to write the protocol and many other clinical trial documents. It should be mentioned that the questions are not in order of importance. Why? Why the research project is taking place? This should lead to the justification of the trial protocol. Who? The core element of clinical research is asking who will be the population of interest. Example, healthy volunteers or subjects with a disease of interest. Who will qualify to participate in the trial is addressed by the inclusion-exclusion criteria. So it is very important to have this clearly defined in the protocol. Other important W is who will be the principal investigator, the members of the DMC, the adjudication committee, the team involved in clinical operation, the safety monitoring board, the biometric team, etc. What? What are the objectives? What are the hypotheses that will be associated with the primary, secondary, and exploratory endpoints? What are the variables that need to be defined according to the study objective and endpoint? Specifically, what are the dependent variables, the independent variables, the factors, covariate, that could help explain the response variable? What are the treatment conditions? This could be the experimental treatment and the comparator arm, if required. If executing a natural history trial, then what are the current treatment to which the patients were exposed during the collection of the natural history data? Another question, what possible issues or event could impact the treatment effect, which is also known as intercurrent events? Along the same lines, what type of medication will be allowed for the study participants to use and which ones will be prohibited to them before and during the trial? And very importantly, what type of assessment will be used to characterize the disease status or progression? What variables are associated with the safety and efficacy profile? Remember that all assessment should use a validated instrument. Once you answer what, you can get specific with which, as in which study instrument will be used for each particular assessment described in the protocol. Again, I use the term instrument to differentiate from assessment, which ought to be used properly calibrated, and again, as I mentioned before, validated instrument with an authorization to its use. For example, let's assume that a suicidal related assessment at baseline in children is part of the planned assessment. Then the instrument to use could be the Children's Baseline Screening version 23 June 2010 of the Columbia Suicide Severity Rating Scale, known as the CSSRS instrument. Notice that I have described the exact version of the tool, which is expected to be used without any modification. Let's look at another example and assume that the assessment is aiming if a person is actively infected with SARS-CoV-2, the virus that caused COVID-19. Then the option could be a nucleic acid amplification test, also known as NAAT, specifically the reverse transcription quantitative polymerase chain reaction, the RT-qPCR test, from a sample collected high in the nasal cavity using a nasopharyngeal swab. Notice that this test, even when validated, does not aim to test for viral infectivity, since for that purpose, a median tissue culture infection dose test, or TCID50, needs to be used. Similar, an antibody or serology test need to be used if the intention is to test if a person likely had a previous SARS-CoV-19 infection? So, in summary, notice that a very unique and validated instrument needs to be used to perform each specific assessment associated with each specific objective described in the research question. All instruments, either an assay, device, a questionnaire, should come with a guideline or set of instructions for its use, specifically if it is using a scoring system to evaluate the response, or if there is a need to convert, for example, the values. It will be crucial to get that information ahead of time, since, as we know, different site and region may use different scales, unit of measurement, reference standard for normal and abnormal, just to name a few. Once again, the W question have no inherent order to them. Now, let's see another example for PK/PD sampling. When the samples will be collected, including if they will be collected pre or post-dose, fasting, fed, sitting, resting, etc. What type of PK/PD lab will be analyzed? Example, plasma, urine? Another question, which PK/PD parameters will be reported? The same applies to questionnaires, as the timing and frequency of the assessment and who will ask and or answer the instrument is of critical importance. Asking which also covers some, for example, biometric questions. Which electronic data capture system will be used? Which statistical method and analysis set are required for the evaluation of the study endpoints? Which sensitivity and supplemental analysis could help understand the impact of deviation from the ITT principle, just to provide an example. Finally, we ask how, not the W in how. This can be asked as a quantity. For example, how many subjects? How long will the study last? Knowing how long the study will last and when the interim analysis will take place is fundamental for study planning. This helps determine the type of data and the level of cleanliness that must be achieved before an interim analysis. How can also refer to the manner in which something will be done. For example, how to handle intercurrent events, meaning describing the intercurrent event strategies that will be considered for each particular intercurrent event. This is incredibly important since intercurrent events could impact the evaluation of each objective and outcome variable. I will address intercurrent events and intercurrent event strategies in detail in a later presentation. But for now, the key takeaway is that there are several intercurrent event strategies to consider. For example, treatment policy, hypothetical, composite, while on treatment, and the principal stratum strategy. Let's continue with the how type of question. How to control for the variability in the variable of interest. For example, outcome response variable, the independent variable, factors, and covariates. That may also impact the interpretation of the effect size and or unbalance the trial. It is important to determine what induces variability. Remember that the higher the variability, the higher the risk for missing the target. Also, high variability results in larger sample sizes. Details such as how the drug will be administered, including the dose regimen, are also of critical importance. How to handle missing doses or dosing outside the allowed window. How to ensure and then measure treatment compliance. In biometrics, knowing how the data will be handled in the clinical trial is fundamental. With all the vendors involved with lab data and any external data captured outside the EDC, we need to know how these data will be transferred to the study repositories and then to the biostatistical team for analysis and reporting, all while ensuring data integrity and preventing accidental unblinding, assuming that the study is blinded. Which then raises the question, how will the blinding and integrity of the trial be maintained? They must be also clearly explained in the protocol and or the blinding maintenance plan, plus the statistical analysis plan. Next, we move to when. This question is often used to determine the timing of clinical operation-related activities. For example, when the trial will be conducted, when will the different protocol-defined assessment be performed. This should be all summarized in the schedule of activities. When will the data be analyzed? When will the data be reported? Meaning, when are the planned interim and final analysis scheduled? When should treatment be stopped? Or even when the trial itself should be stopped, known as stopping rules, which could be based on safety and or efficacy predefined criteria, and so on and so forth. Where helps answer questions like where, from a geographical perspective, the study will be conducted. For example, where the sites are located, which countries, where the study visit will take place, clinics, virtual visit, etc. The investigators should be also asking if there is any expected difference in the outcome variable in terms of the location or geographic regions. If so, it may be necessary to adjust the analysis based on region or site.
At this point, you may think you have a good grasp of the necessary questions to address the research question. And though we've provided several examples, keep in mind there are still many other questions that could arise during protocol development. Protocol development requires a multidisciplinary team, and everyone involved will be asking their own questions to assemble a robust protocol and avoid protocol amendments.
In our attempt to find the links between ICH E9R1 and the scientific method, let's look at the FDA drug development process, specifically Step 3, which focuses on clinical research.
Before we continue, do you notice the W questions in the FDA document? This confirms that the process for operationalizing the research question is fundamental in clinical trials. Importantly, this process must occur before the trial begins and prior analyzing the data. Remember that amending the protocol or SAP after database lock is not allowed, especially once the data is unblinded, as this would introduce bias and compromise data integrity, which would be a major concern.
Moving back to the scientific method from a clinical research perspective, let's go over drafting, formulating the study hypothesis. So, what is a hypothesis? A research hypothesis is a statement of expectation or prediction that will be evaluated, probably tested, once the data is collected and analyzed using validated processes and predefined plans by a group of qualified people. It could be also defined as any concept or supposition which is used to organize a group of facts in order to facilitate further research. In general, if a given hypothesis conforms with a provided fact, then it is said to be verified or true. Likewise, a fact which agrees with a hypothesis is said to be explained by that hypothesis. Hypotheses can come in different types: working hypothesis, descriptive, divided in relational or causal, the null hypothesis, and the alternative hypothesis, which could be divided also as directional and non-directional hypotheses. A working hypothesis is a hypothesis that is provisionally accepted as the basis for further research, in the hope that a tenable theory will be produced, even if the hypothesis ultimately fails. In this way, a working hypothesis is an accepted starting point. In essence, the working hypothesis is used to engage with problems previously undefined by means of both theorization and experimentation. I recommend checking out the link below for further details. From Samuel Taylor, a publication from 2021. Descriptive hypotheses are used to describe the existence, characteristics, size, distribution, or attribute of a particular variable, since they usually contain some characteristic value of that which you want to observe or evaluate. They are especially useful when you want to learn more about the object of investigation. The variable of interest can represent anything: an object, a person. It could be an organization or even an event. For example, your descriptive hypothesis could be related to the safety profile of the investigational medical product and could be written as: what is the difference between treatment arms in the incidence of treatment emergent adverse events after drug administration up to 30 days after the last treatment? Once your data is collected, a summary table and listing describing those events using descriptive statistics would be produced, and no formal inferential hypothesis testing would take place. A relational hypothesis is used when we want to explore a possible relationship between a set of variables. For instance, the relationship between age group and a particular disease. In this type of hypothesis, there isn't a clearly defined dependent or independent variable, since it is basically an exploratory investigation trying to define possible correlation or level of association for further research. If the idea is to measure, for example, how changes in one variable affect the other variables, and there is a distinct definition of dependent and independent variables, then, in order to evaluate this causal relationship, a causal hypothesis can be written. Causal hypothesis could be formulated to determine the direction and magnitude, or both direction and magnitude, of a possible relationship. For example, using correlation analysis to determine how level of A1C in hyperglycemia itself changes in vascular function and structure and hypertension. Or, for example, is the increase in new COVID-19 symptomatic patients related to the number of people fully vaccinated? The null hypothesis is a statement that assumes no relationship exists between a set of variables. If the hypothesis significant testing, the null hypothesis generally denies whatever we define and or trying to test in our working hypothesis. It is important to note that although the goal is usually to disapprove the null, one can only reject or fail to reject the null hypothesis. Failure to find sufficient evidence to reject the null hypothesis does not imply the null was accepted, proven, or true in any capacity. The alternative hypothesis is essentially the opposite of the null hypothesis. When running hypothesis significant testing, the alternative hypothesis, also known as H1, can be accepted if and only if there is sufficient evidence, data, to reject the null hypothesis, and a predefined alpha level, which is also known as the type 1 error. The alternative hypothesis is usually associated with an expected effect size, delta, which could be expressed as a difference in proportion, an odds ratio, a difference in means between treatment arms, etc. A type 1 error, false positive, occurs if an investigator rejects a null hypothesis that is true in the population. Thus, the type 1 error rate, alpha, is a probability of rejecting the null given that it is true, and is usually set at the 0.05 alpha level in a two-sided test or at 0.025 alpha level in a one-sided test. If the investigator fails to reject a null hypothesis that is false in a population, it is called a type 2 error, a false negative. The probability of this occurrence then is called the type 2 error rate, beta. One-sided tests are called directional tests since they allow us to make claims about superiority, equivalence, or non-inferiority by a defined margin. A non-directional hypothesis takes the equal sign and assumes that there is no difference in the null hypothesis, or that the difference is not equal to zero in the alternative hypothesis. But in reality, in most cases, the analysis aims to detect a difference in where the absolute value favors the experimental arm. Let's talk now about the study aim, objective, endpoint, the scope, and the limitation of a clinical trial. The study aims is the overarching purpose or goal of the trial. This could be something like to evaluate the safety and efficacy of the experimental treatment versus placebo after X time of treatment administration in a particular patient population. But be careful. Do not confuse the study aim with the primary objective. The objective should be clearly defined target within the study aim and are specific enough to be measured with a validated instrument. A well-written objective uses action verbs such as to compare, to calculate, to assess, to determine, to verify, or to describe. In clinical trials, we commonly see the following type of objective. Primary objective, driven by the primary hypothesis, and that is the main goal of the study. Sometimes it could be to evaluate the safety or to evaluate the efficacy profile. The secondary objective. Any other objective that will be evaluated, tested, will be considered as a secondary objective. Secondary objectives are usually planned to support the primary objective and could be considered in the label of the experimental drug if there are enough evidence to accept the alternative hypothesis associated with it. In theory, the study should be powered not only to detect statistical significant differences around the primary objective, but also for all secondary objectives as well. To avoid the inflation of the type 1 error, a formal testing strategy should be defined to explain how those secondary objectives will be tested. Presenting p-values for any analysis without proper adjustment for multiplicity could be technically allowed, but the investigator must explain that those are nominal p-values and the result should be considered descriptive in nature. In other words, nobody reading the result should mistake the result as actually being statistical significant since the adjustment for multiplicity was not performed and the underlying assumption of a given statistical model have not been properly taken into consideration. Exploratory objective. These are also exploratory in nature and include any additional analysis intended to help further understand the efficacy, safety, PK, PD, or PG profile of the investigational drug. As a general rule, do not collect any data if there is no plan to analyze it. When writing the objective, it is imperative to be very clear and include all the key attributes. For example, the population of interest, the treatment conditions, the outcome variable, the main intercurrent event that could impact the interpretation of the endpoint associated with the objective, the instrument that will be used to collect the outcome variable, and of course, the timing for the evaluation. Let's look at this example. To assess the efficacy of two IMP, investigational medical product, induction regimens, high versus low dose, without the use of rescue medication in achieving clinical remission at week 12 in subjects with moderately to severely active Crohn's disease and evidence of mucosal ulceration at baseline. Notice, in blue, the treatment conditions. In yellowish, the key intercurrent event. In black bold, the outcome variable that could take a yes or no. And in red, the population of interest. Very important, I use a superscript to define the variable of interest and the instrument that will be used to capture the effect size at the patient level. For example, as measured by the Crohn's disease activity index score, the CDI, and the condition is less than 150 at week 4, notice the timing, and an endoscopic response defined as decrease in simple endoscopic score for Crohn's disease, the SESCD, with a borderline greater than 50 from baseline, or baseline score using the same instrument of 4 and at least 2-point reduction from baseline. By the way, this is what is called a composite objective. To emphasize my previous comment about endpoints, let's look at the FDA endpoint definition. Principal indicators used for assessing the primary question, example, hypothesis of a clinical trial, a variable that pertains to the efficacy or safety evaluation of the trial. An endpoint is more specific compared to an outcome variable. since the endpoint relate to the plan objective of the study. And this is coming from the FDA Glossary of Terms of Clinical Trials for Patient Engagement Advisory Committee meeting. The confusion arises when the general term variable is introduced without making emphasis that the variable should capture the value of the actual comparison to either accept or reject the null hypothesis. Another issue with this definition is that it limits its use to the efficacy or safety evaluation. But what about PK, PD, pharmacodynamic, or something else? In general, safety and efficacy endpoints ought to be clinically meaningful. They must actually provide some relevant clinical benefit for the population of interest. But remember, not every endpoint, like the one measuring biomarkers, is inherently clinically meaningful. For example, a patient might have shown a rapid rate of viral elimination, but it will be hard to pinpoint as clinically meaningful if it is not associated with an actual endpoint like reducing the need for another critical medical procedure like surgery or something else. Let's look at this example of how an endpoint could be written. The difference in proportion, which is delta, between treatment arms, which refers to the treatment condition in subject with moderately to severe active Crohn's disease that have achieved clinical remission. I'm going to stop here for a second because that clinical remission will become the variable of interest. Then in the bottom, I can use the superscript to explain the definition. Now, there is another component in this example which is critical. Per central reading is specifying how that clinical remission and how those evaluations from the CDI and the SES-CD score will be captured and what kind of data will be used to define clinical remission. In this case, they're defining per central reading. And then, very important, at week 12 in the induction phase, this is the timing. In summary, a very well-written endpoint refers to delta, the treatment effect, which is going to be computed and estimated using a particular population-level summary, a point that I will explain also later. In general, there are two types of endpoints: direct endpoints, which are meant to be clinically meaningful, and surrogate endpoints, which are somehow related to a particular clinically meaningful endpoint. There are cases in where objective could imply co-primary endpoints, in which adjustment for multiplicity and a testing strategy must be in place. In other cases, composite endpoints could be used, which is a type of endpoint with multiple sub-endpoints and must be achieved in order to declare efficacy or safety, like the Crohn's disease example that I showed before. The scope provides details about the study duration, the type of trial, whether it is a parallel, crossover, randomized, the type of control arm, etc., the duration of the study phases, and the actual procedures, assessment, and other specifications that will be covered within the protocol. Again, the study must define what will be done, in what settings the assessment will be done, for example, in a clinic or a virtual visit, when those assessments will be performed, and how many participants will be required, how long the participants will be treated. The protocol should also provide details about the safety follow-up period, use of rescue medication, use of prohibited medication, potential extensive use of the experimental treatment after the trial is over, and availability of resources to access the clinical site in case that is required. Other details include, for example, who will be allowed to participate in the clinical trial and how eligibility will be determined. To understand the limitation of a protocol, just think of what isn't in the scope. From a subject perspective, it could be who will not be eligible to participate in the study, which could be described in the exclusion criteria. Other limitation could be which site or geographic region the study will take place. The inclusion of one set of participants may be inherently excluding others. Also, what will not be done in the study. One aspect that I would like to emphasize is the availability of the study treatment once the subjects complete the study, since in some cases they may expect to continue treatment, especially if they observe clinical benefits, but the investigational product is not available in the market. In this situation, an open-label extension study may be planned or not, but either way, those details need to be specified in the protocol.
Now that we've gathered all this knowledge, we can begin to describe the link between the scientific method and the Estimands framework, or at least with some of its components of the former one.
For this part of the discussion, we're going to be using E9R1, a statistical principle for clinical trials, addendum, estimates, and sensitivity analysis in clinical trials, guidance for the industry, published in May 2021. Let's dig deeper with a very critical constructed mindset and check the definition of the estimate, a precise description of the treatment effect reflecting the clinical question posed by the trial objective. It summarizes at the population level what the outcomes would be in the same patient under different treatment conditions being compared. Let's see how the W's could be mapped to this definition. The estimate of theta is defined as the target of the estimation, or the quantity that is being estimated for a population. Notice, in this example, the difference in proportion refers to the target of the estimation, and that will be computed between treatment arms in subjects with moderately to severe active CD that have achieved clinical remission per central reading at week 12 in the induction phase. So clinical remission here refers to the outcome variable, and the difference in proportion is the target of the estimation that will be later mapped to the hypothesis or the primary hypothesis in the clinical trial. According to the Estimands framework, there are 5 attributes of the estimates, and I am trying to do my best to map it to the W's from my previous slide. The what, associated with endpoints, treatment condition, and intercurrent events. The who, associated with the population of interest, and the which, the population level summary. Let's see the mapping between the Estimands framework and the what, and what we discussed earlier in the slide 7 around the scientific method. In ICH E9, they describe a variable or endpoint, making no distinction between the two, the outcome variable, and they define the variable or endpoint which can be obtained from each patient, and that is used to address the clinical question. The specification of the variable may include whether the patient experienced an intercurrent event. But notice again, in the scientific method, we are talking about the research hypothesis. The point is around delta and showing whether or not we can reject or not the null hypothesis. A better approach could be, for the interpretation of ICH E9, it could be correct to assume that the variable of interest is the outcome variable. That's correct, but not the endpoint. And then if we just remove our endpoint in that definition, that could be more appropriate. In that way, like I mentioned before, the synchronization and the link between the ICH E9 and the scientific method will be almost perfect. Let's explain it with an example. And I'm going to be using the same data from the Crohn's disease trial that I presented before. Let's check carefully. The comparison group during the induction phase using a standard induction dose and then using a higher dose, and it was a superiority test. The p-value that ended up now being statistical significant, and they make some comment about some adjustment that they did for previously used infliximab. Also, pay attention not only to the statistical method, the CMH method, but to the result. The estimate associated with the estimate, right, is 0.3 for the risk difference between the investigational medical product higher dose and the induction phase and the standard induction dose. So again, the variable and the endpoint cannot be confused because one, the variable is associated with the patient level outcome, and delta is associated with the hypothesis that are being tested during that particular clinical trial. To conclude, it is important to distinguish between the outcome variable, y, and the endpoint associated with the hypothesis defined in the research question, which at the end is the quantity being computed, delta, the estimate of the treatment difference. We must define the conditions in which the outcome variable and the endpoint will be analyzed. Remember, the estimate attributes are not limited to intercurrent event, population of interest, treatment condition, and population level summary. A more accurate operationalization of the research question demands other critical attributes to be defined. For example, the instrument to measure the outcome variable, the study duration, just to name a few.
If you enjoyed today's topic, make sure to hit like and subscribe and hit the notification bell to get updates whenever we post new videos. With that said, we want to thank you for joining us for this presentation, the first in a series on ICH E9R1, protocol development, and statistics in clinical trials. Until next time, get correlated.